divide? Well, living things grow by producing more cells, not because each cell increases in size, so we need to replace them. Especially when there's a injury or we need to repair damaged tissue, we need to create more cells. If the cell is too big, it cannot get enough nutrients into the cell, so we need more cells that are of the same size. Just a fun fact, the human body makes more than 20 billion new cells every day. Over the course of 7 years, all body cells have been replaced, except for nerve cells. So how does our body make all of these new cells? This is a study of mitosis, but this is the new vocabulary that we're going to be learning. So let's start with a chromosome. So a chroma means colored, and soma is just the body. Whenever scientists are looking for chromosomes, this is called karyotype. This is what they're looking for. So these structures are found in the nucleus, and they all contain DNA. So this is a chromosome, as we can see here. And the arms of a chromosome are called the chromatid. So here are the chromatids. The centromere is the center of the chromosome where the chromatids are connected. And why are chromosomes so important? Well, they contain genes, and like we saw in a previous class, genes makes proteins. So here we can have a gene, in a different gene, in a different, all of them coding for something different. For example, maybe this one is about the hair and how it looks like, and maybe this other gene is about the eye color. So every gene codes for something different, and like we saw in DNA, transcription to RNA and then translation to protein, this is the code of life. So it's just a sequence of DNA that codes for a protein and determines a trait. So these are like the color bands on a chromosome. Proteins, so make up the traits, characteristics of living things. And where do we find these proteins? Or who detects the proteins? It's the genes on the chromosome that we need to undergo all those processes. Okay, the centrioles are small structures located outside the nucleus but near the nucleus, and they create the spindle fibers. And the spindle fibers are fibers formed by the centrioles, and they're going to start pulling the chromosomes apart whenever this is ready. So here are these centrioles, the star-looking shapes. And these fiber-looking things are the spindle fibers. So these fibers are the ones that are going to start pulling the chromosomes apart when this is ready. And so mitosis. As we can see in this picture, we are about to get two cells. They're about to split. Here's one nucleus, here's this nucleus, and all these fibers are the spindle fibers from the previous slide. Mitosis takes place in body cells, every cell except for the egg and the sperm. The first phase of mitosis is called prophase. And during prophase, the duplicated chromosome condenses, so it means that it becomes more visible. It's short and very visible. Before, it was in the form of chromatid, it was just all over the place. But in prophase, they start taking shape. This is more efficient, so the cell can divide faster. So here's just prophase. It's getting ready for the process of mitosis. The first phase, the chromosomes are very visible. We can see the centromere here. holding the chromatids. Here are centrioles, the spindle forming. Everything is getting ready. The centrioles moved to the opposite sides. We want to create two cells. So they're moving to opposite sides to help organize the spindle. The spindle forms, all these fibers form, and DNA strands attach at the centromeres. They want to start pulling them apart. Of course, at this point, we're going to start breaking the nuclear membrane. We want to break this envelope. And now the spindle fibers are able to come in, attach to the centromere, and start pulling. Before that, we have our next phase, which is called the metaphase for middle. Metaphase for middle. So during the metaphase, the centromeres of the duplicated chromosomes line up in the center of the cells. It's in the middle. They're lining up. They're getting ready to be pulled. You can see the spindles are here in the centromere. The spindle fibers connect to the centromere of each chromosome to the two poles. So one is going to start pulling one chromosome apart, and this one is going to be pulling the other one on the opposite side. Anaphase is our next phase. Here is the third phase of mitosis. The centromeres are pulled apart and the chromatids separate to become individual chromosomes. So here, they're pulling on the centromere, pulling on them, and now we have one chromosome on one side, the other chromosome on the other side. So it's going to do this with all of our chromosomes. The chromosomes separate into two groups near the poles. As you can see, we can easily see how we're about to get two different cells out of this one. In telophase, the fourth and final phase of mitosis, the chromosomes spread out into chromatin. So instead of having this shape, they're going to start to uncoil a little bit. The nuclear envelopes start reforming. We almost have two complete cells. So this is the end of mitosis with almost two complete cells. A nuclear envelope reforms and the spindle breaks apart. So they are detached from the centromeres. The nucleus becomes visible in each daughter nucleus. Now we have one nucleus, one nucleus. Now, that was the end of mitosis. Next, we have something called the cytokinesis, which is when we divide and split the two cells. It's different for both animal and plant cells. In animal cells, the cell membrane is drawn until the cytoplasm is pitched into two equal parts. So here's where we get our two true daughter cells. Each part contains its own nucleus and its own organelle, cell membrane, everything. So at the end we have two daughter cells. So this was the end of mitosis. This is cytokinesis. We've split it and we have two daughter cells, identical daughter cells. In plants, it's a little bit different. Remember that in plants we have the cell wall. So the cell membrane is not flexible enough to make this cut because the cell wall is very rigid. So instead, we're going to have a cell plate to form between the dividing nuclei, and the cell wall forms in the middle of them, like this. We have almost everything ready for during mitosis, at the end of mitosis. So what's left for cytokinesis? We form in the middle something called the cell plate that becomes the new cell wall. So we don't have to actually cut and split it. We just form the cell plate that becomes the new cell wall. Here is the picture of how everything looks like under the microscope. Here we have prophase. Chromatin is becoming more visible and shorter. Everything is getting ready for that division. We have the centromeres and the spindle starting to form. And then in metaphase, all of them align in the middle. The spindle fiber connects to the centromere to start pulling on them, and this happens in anaphase. They start to pull away from each other. And then in the telophase, the daughter nuclei are formed, everything is almost complete, but they're not two separate cells yet. We need the next phase, which is called cytokinesis, where we actually split it. Now, in bacteria, this is different. Bacteria do not have a nucleus in prokaryotes. So the process, we don't call it mitosis in bacteria or prokaryotes, we call it binary fission. So the chromosome replicates, and the two daughter chromosomes, they just move apart. So here is the chromosome. We get a new copy. It starts to replicate. Replication continues. Here we have the two copies. And then a new plasma membrane grows inward. And then they split. So we don't call this mitosis. This is just called binary fission. Okay, so let's see if we can identify them. Click pause, write down your answers. When you're ready to check them, then click play again. Okay, for this one, you can see in anaphase, they start to be pulling away. A for anaphase, A for away. All those chromosomes are started to go into each side, each end of the cell. This one, it's almost ready. We just need to make that cut. This is called the telophase. This one, we already can see the cell membrane. It's done, it's ready. When mitosis and cytokinesis, which is the last phase of cytokinesis, the splitting and the formation of new cell membrane. In here, in this one, the chromosomes are not very visible, but they're getting there. So this is the interface. In here, we can see how they align in the middle. So this is the metaphase, M for middle. And the last one, they start to condense, shorter, more visible. This is the prophase. Compare the prophase to the interface. So this was before with all the chromatin. Here we can clearly see, or more likely see, the chromosomes. Okay, so mitosis only occurs in body cells or somatic cells. Is another name for them. It doesn't occur in eggs or sperm. The two new cells have the exact same number and kind of chromosomes as the original. Here is the parent cell, what we started with. Then here's undergoing DNA replication. We duplicate each chromosome. And then at the end, we get two daughter cells exactly like the parent cell.